In Lab 7, we're going to be looking at bacterial endospores and bacterial motility. Both of these are topics, of course, we covered back in Unit 1 when we discussed bacterial anatomy. Now, we saw that a few genera of bacteria in the domain Bacteria, namely the genus Bacillus, the genus Clostridium, and the genus Clostridioides, have the ability to produce resistant survival forms termed endospores. And they're not reproductive, but rather survival in nature. These endospores are resistant to heat, to drying, to some radiation like ultraviolet radiation, and a variety of chemical disinfectants. The process of producing endospores is called sporulation. And we have a couple of animations down here I've included that demonstrate endospore formation or sporulation if you want to review those. But eventually, each single vegetative bacterium, a vegetative bacterium being either one that doesn't have endospores or one that's in the process of producing endospores, produces a single spore within the bacterium. Once the endospore is formed, the vegetative portion of the bacterium, the remainder of the bacterium, is degraded and the dormant endospore is released. Sporulation generally takes about 15 hours. And here's a nice electron micrograph of an endospore Bacillus thermophilus. So this is just the endospore that was formed inside the bacillus. And the endospore consists of a small mini dehydrated bacterium containing a chromosome and some ribosomes, surrounded by a thick protective coat called a cortex, composed mainly of peptidoglycan, and then a spore coat composed of proteins and sometimes an exosporium. And of course, that endospore can survive under harsh environments pretty much indefinitely until there's a suitable environment, in which case the endospore germinates and a single vegetative bacterium emerges. And in this illustration, or this photomicrograph, we see an endospore of Clostridium sporogenes germinating. So these are endospores in figure 1N that haven't germinated. Here's one that is germinating, and you see the vegetative bacillus emerging from the endospore and already undergoing binary fission. Now, the endospores themselves are harmless. It's only the vegetative bacteria, if it's a pathogenic endospore producer, that is able to cause the infection. But the endospores are often involved in the transmission of some diseases to humans. We'll be talking about each of these diseases later on in Unit 3 when we get to bacterial pathogenicity. But examples of diseases that are caused by endospore producers include anthrax caused by Bacillus anthracis, tetanus caused by Clostridium tetani, botulism caused by Clostridium botulinum, gas gangrene caused by Clostridium perfringens, and antibiotic-associated pseudomembranous colitis caused by Clostridioides difficile, that used to be called Clostridium difficile. Now, because of the resistant nature of endospores, they are rather difficult to stain. We have to use strong dyes, vigorous staining conditions like heat in order to stain the endospore. But then they're also hard to decolorize afterwards. And so the procedure we see next is the procedure we normally would have done if we were doing endospore stains in class. However, in this endospore procedure we're showing you, once the slide has been heat fixed, it's then has a piece of blotting paper put over it and it's saturated with malachite green, and then that slide has to be steamed for 5 minutes over boiling water. Now, before we moved to our new building, we used Bunsen burners to boil the water so we could do endospore stains. Of course, we don't have gas in our current labs. We use the Bacto incinerators for aseptic technique instead. And we don't have enough hot plates for us to actually do the endospore stain in lab. So what we're going to show you is a demonstration slide of what this endospore staining procedure would look like. In this case, we're staining Bacillus megaterium, a common soil bacterium, which is a streptobacillus. So in this staining procedure, if we were to do it as we see in figure 2, the vegetative bacteria that haven't produced endospores yet, like we see here in figure 2, stain red. The remainder of the bacterium, the vegetative bacterium other than the endospore, stains red, but the endospores themselves stain green with malachite green. The red is of the vegetative cells due to the dye safranin. So we see this as a streptobacillus, and each bacillus eventually makes a single endospore. Here's a nice little chain of 2 where we see each bacillus has produced a single endospore. Once the endospore is completely formed, the vegetative cells degraded and the endospores are released. We're also going to be showing you a demonstration slide of Bacillus anthracis, the bacterium that causes anthrax. This too is a streptobacillus. Here we see some vegetative cells, which with this procedure stain blue. And then the remainder of the cell that isn't the endospore, the rest of the vegetative cell also stains blue. In this case, the endospores appear colorless. So here we see a chain of bacilli, and each single bacillus has produced a single endospore. And again, once those are fully formed, then the vegetative cells are degraded and the endospores are released. We're also going to show you a demonstration slide of Clostridium tetani, the bacterium that causes tetanus. We see that in figure 4. So in this case, the vegetative cells stain kind of a gray-blue color, and the endospores are colorless. So here's a vegetative cell that hasn't made endospores yet, and here are several that have produced an endospore. So you see this is a single bacillus. Each bacillus makes one endospore. Now, one of the unique things about the genus Clostridium is that many of the species, when the endospore forms, produce an endospore that's larger in diameter than the rest of the bacillus, giving the bacterium a characteristic tennis racket shape when viewed under the microscope, or sometimes called a club shape. And we don't have a slide of this to show you, but we have a picture online showing the endospore stain of Clostridium botulinum using the same staining procedure that we used on the Bacillus megaterium up in step 1. We see that in figure 13, where again the vegetative cells stain red and the endospore stain green. And again, we see that nice tennis racket shape or club shape of the genus Clostridium. So on any of these slides, when you see them on the practical, you should recognize the unique structure we're looking at there as an endospore. And if we see that tennis racket shape, we should identify the genus as Clostridium. And again, remember, genus is the first of the 2 scientific names. We don't want the species name, just the genus name. So those are bacterial endospores. The second part of the lab deals with bacterial motility. And we know that many bacteria are capable of motility, or moving under their own power. And when swimming in a liquid, the motile bacteria propel themselves using flagella. Now, as we saw in Unit 1 in lecture when we talked about flagella, the filament is the first part of the flagellum, the rigid helical structure that extends from the cell. This is composed of the protein flagellin, and it's arranged in helical chains to form a hollow core. That in turn connects to a hook, which is a flexible coupling between the filament and the basal body. And the final part is called the basal body, which has a rod and a series of rings that anchor the flagellum in the cell wall and the cytoplasmic membrane. And that connects finally to a little nanomotor or rotary motor that enables the flagellum to rotate, typically either clockwise or counterclockwise, to propel the organism through the surrounding fluid. And figure 5 is the illustration we showed you of that. So the filament up here is a hollow protein tube that's helical when you see it sticking out of the bacterium, composed of flagellin. That connects to a flexible coupling called a hook, which attaches to a ring or a rod, and then there are rings around the rod that anchor the bacterium in the cell wall, and that connects eventually to the motor down here. And this is propelled by electron motive force. As protons or hydrogen ions are pumped across the cytoplasmic membrane by the electron transport system, they accumulate between the membrane and the wall. And when you have a sufficient gradient of these positively charged hydrogen ions, they re-enter through these Mott proteins, and that provides the energy, the moving protons that rotate the motor, causing the flagellum to rotate. We also learned that bacteria have a number of arrangements of flagella, and that can be helped in their classification and identification sometimes. So the arrangements we learned are, as we see in figure 6, monotrichous, a single flagellum at one pole; lophotrichous, two or more flagella at one or both poles; amphitrichous, a single flagellum at both poles; paratrichous, where the organism is surrounded by flagella. So here we see some photomicrographs of these bacteria. Figure 7a shows you a Vibrio species that has a monotrichous arrangement. So here we see the little curved Vibrio with a single flagellum. There's another one up here, another one here that has a flagellum. So you'd recognize that as a monotrichous arrangement. An amphitrichous arrangement is a single flagellum at both poles. And the spirillum we're going to show you today has an amphitrichous arrangement, a single flagellum at both poles. So you'd recognize that arrangement as amphitrichous. Another bacterium with an amphitrichous arrangement, the next one is lophotrichous. And a lophotrichous bacterium has two or more flagella at one or both poles. And we see that in figure 8. So this is a different spirillum that has a lophotrichous arrangement. We see a bundle of flagella, in this case at both ends. And another bacterium that has a lophotrichous arrangement is the bacterium responsible for ulcers, called Helicobacter pylori. And we'll load that illustration in. So this is an electron micrograph where we're going to see the lophotrichous arrangement of Helicobacter pylori. This is a kind of a spiral-shaped bacterium with a bundle flagellum at one end. A paratrichous arrangement is where the flagella completely surround the bacteria, as we see in figure 9. Now this is Proteus vulgaris, one of the bacterium we'll learn about later on in lab 12, which has a paratrichous arrangement. Bacteria like E. coli also have a paratrichous arrangement. Now we're actually seeing two forms of the Proteus here. You may remember from lecture that some bacteria also have a swarming motion, which they use when they're on a solid surface. And what they do in their swarming form is that the bacteria elongate and produce many more flagella. So this is actually a swarming form of Proteus that has the paratrichous arrangement. And these are the typical Proteus, the non-swarming forms. But again, you can see they're shorter, they have fewer flagella, but it's still a paratrichous arrangement of flagella. So you'd recognize that arrangement as paratrichous. And then one group of bacteria, the spirochetes, have internal flagella called axial filaments, or sometimes endoflagellum. And these originate from a basal body, and then the axial filaments wrap around the spirochete from the end towards the middle, from both ends. And they're located above the peptidoglycan cell wall, but underneath the outer membrane, like we see in figure 10. So this is an illustration where part of the bacterium is broken away. There's the insertion structure, or basal body with a hook, and notice the filament of the flagellum wraps around the spiral, starting at the end going towards the middle from both ends. But notice it lies on top of the peptidoglycan, but underneath an outer membrane. So those are internal flagella, or axial filaments, seen only in spirochetes. We also saw that flagella can play a role in bacterial pathogenicity. For example, the mucosal surfaces of the respiratory tract, the intestinal tract, and the genitourinary tract constantly flush bacteria away to prevent colonization of the host mucous membranes. but many motile bacteria can use their motility and chemotaxis, or they swim towards an attractant in this case, to swim through the mucus towards the mucosal epithelial cells in order to make contact with those cells and eventually colonize. So we showed you this animation. So again, here we see a bacterium with a flagellum, a monotrichous arrangement. This is mucus, the pale yellow, and this is a gradient of chemoattractant. So the bacterium is going to swim towards increasing chemoattractant, which are chemicals produced by the epithelial cells, until they make contact with the cells, and that way they can resist being flushed out by the mucus. So here we see the bacterium using its motility and chemotaxis to swim through the mucus to make contact with mucous membranes, such as those in the gastrointestinal tract or in the bladder. We also saw that because of their thinness and their internal flagella or axial filament, their corkscrew shape and their motility, spirochetes are more able to penetrate host mucous membranes and skin abrasions and enter the body, but they can also use their motility to enter lymph vessels and blood vessels and then be carried elsewhere in the body, where they leave the lymph vessels or blood vessels and set up an infection at a secondary site. So the spirochetes often use their motility to disseminate to other body sites, such as Treponema pallidum, which causes syphilis, Leptospira that causes leptospirosis, and Borrelia burgdorferi that causes Lyme disease. And here's the animation we showed you of that. So in this case, the spirochete uses adhesins at its tip to bind to the surface of cells. It produces invasins that soften up the membrane of the endothelial cells, in this case forming a capillary, so they can actually drill themselves through the cell and enter the capillary and be carried by the blood. They can also use our motility to pass between cells, and we see both of these in this animation. So there's the bacterium drilling through the cell so it can enter the capillary. That one passed between the cells. And as the blood carries them elsewhere, they can use the same mechanism to leave the lymph vessel or the capillary of the blood vessel and set up a secondary invasion at another site. Now, there are several ways we can detect bacterial motility. One is direct observation with a microscope. And although we can see motile bacteria with a conventional light microscope like we use in lab, we can't see them very well because we're looking at living unstained bacteria, and they're very difficult to see without staining. And all the dyes we use kill the bacteria where they stop swimming. But there are special purpose microscopes that use special optics to give you the effect of staining without actually having to use dyes. And the first of these is called a phase contrast microscope. Now, this uses special phase contrast objectives and a condenser to control the illumination to give you the optical effect of a direct stain. It causes the organism to appear darker than the background. So the thicker a specimen is in the liquid, the brighter it appears. The background appears bright and the organism appears darker against that background. So we normally would set this up under our phase contrast microscope in the lab, but these days there's so many good videos of that on YouTube that you can actually see it better there. So this is a link to a phase contrast microscope showing you motile Pseudomonas. And we'll load that video in. So notice here that when the organism is in focus, it appears darker than the background. And when the organism is not in focus, then it appears light. So notice it swims in and out of focus, but notice we see tumbling motility, where the organism, there's a nice run we just saw, we see tumbling, and that's how it, of course, changes reaction. So when bacteria are actually swimming, you see them moving very rapidly frequently, often many times their length a second, in runs and tumbles, which they use to eventually obtain a more optimum environment. So with the phase contrast microscope, the organism appears darker than the background, but it's done with optics. A dark field microscope uses a special condenser to direct light away from the objective lens. So in this case, the background is going to appear dark and the organism will appear bright against a dark background. And in figure 11, we see the spirochete Leptospira. This is a photomicrograph I took decades ago when I worked with Leptospira. And you can see how the organism really pops out in a dark field microscope. The spirochetes are so thin, they're very difficult to see. But under dark field, you can see them very clearly because the organism appears light against a dark background. So that's kind of like an indirect stain, but being done with optics rather than dye, so we can observe living organisms. And here's another photomicrograph of Borrelia burgdorferi that causes Lyme disease. And again, you can see the spirochete, the very thin spirochetes, very clearly against the dark background. And we showed you a little movie of that back in lecture. It's this link right here. We'll load that in, where you can actually see pretty nicely the corkscrewing motility of the spirochetes. So this is a movie taken with a dark field microscope. And again, you can see the little corkscrewing motion of the tip of the spirochete as it translates through the medium. This is actually an ad for something called Cytoviva, which allows you to see organisms even more brightly under a dark field microscope. But again, you see the spirochetes very clearly and the little corkscrewing rotation of the tip. Now, a second way we can detect bacterial motility is to use motility test medium. And motility test medium is a semi-solid agar. It's a stab tube, which we inoculate by stabbing, as we learned in Lab 2. It is solid enough that it forms a soft gel so that non-motile organisms can't move away from where it was stabbed, but a motile organism can swim through the gel and swim away from the stab line. So when a non-motile bacterium is stabbed into motility test medium, growth of the bacteria occurs only along the line of inoculation. The growth along the stab line is very sharp, very well defined, but it doesn't swim away from the stab line because it's non-motile. And we see an example of that in figure 12A. So the motility test medium has kind of a reddish color. There's a dye added that turns red in the presence of oxidation reduction reactions. So the more bacteria you have growing at a particular location, the darker red the agar appears. So we see very heavy dark red growth along the stab line. This is where the stab of the loop went into the agar, but notice the bacterium hasn't moved away from the stab line. There's very sharp, well-defined growth along the stab line, indicating that this is a non-motile bacterium. Now, on the other hand, if the bacterium is motile and you stab the motility test medium, it's going to swim away from the stab line. So you see growth radiating away from the stab line out into the agar itself from the line of inoculation. And the growth along the stab line is much more cloud-like or billowy. You can't see the stab line really well. And we see an example of that in figure 12B. So notice in this case, the agar was stabbed down the center to here, but now you can't see the heavy growth of the stab line. We see the bacteria are swimming out into the agar from the stab line, and as they replicate, it gets darker and darker. So again, in a motile bacterium, the bacteria swim away from the stab line and grow out into the surrounding motility test medium. And we'll be trying motility test medium in lab today. And the third way to indirectly test motility is to look for flagella, using flagella staining. So we assume that if we see flagella after flagella staining, the organism must have been motile when it was alive. But flagella themselves are very thin. They're only 20 to 28 nanometers in diameter. They're below the limits of resolution of a normal light microscope. When we looked at the Pseudomonas up here under phase contrast, which we'll show you again in a minute, you didn't see any flagella because they're too thin to see with a conventional light microscope. Of course, we can see them with the higher resolution electron microscopes. So what happens in flagella staining is that we treat the flagella with special dyes and mordants that build up as layers of precipitate along the length of the flagella, making them visible under the microscope. But that's kind of a delicate staining procedure. There's a bit of an art to that, so we're not going to bother with that in lab, but we will show you several demonstrations of flagella stains. So for the motility test part of the lab, we have several things we're going to be looking at and 2 tubes we're going to inoculate. We're going to observe the phase, we're not going to actually observe the phase contrast microscope, but again, we'll show you that YouTube movie of modal Pseudomonas under phase contrast microscopy. So again, if we look at that video that I showed you just a few seconds ago, we'll load it in again. We can see the bacteria swimming very nicely. And again, when it's in focus, the organism appears darker than the background under phase contrast. And we can see the runs and the tumbles and the changes in direction of bacterial motility, and we can see they can move quite rapidly. Pseudomonas, for example, swims about 50 micrometers a second, and it's only about 2 micrometers long. So that shows you what motility looks like using phase contrast microscopy. Again, we won't set up the microscope, we'll just show you this YouTube video. But again, notice we can't see the flagella because they're too thin to see with a conventional microscope. But we see the results of the flagella, the actual motility. We're not going to bother with a dark field microscope demonstration, but I did show you a couple of photomicrographs of spirochetes under dark field, where the organism appears lighter than the background. And then in lab, we're going to be inoculating 2 tubes of motility medium per pair. We're going to stab one with Pseudomonas aeruginosa and the other with Staphylococcus aureus. Now note the caution up here. Pseudomonas aeruginosa and Staphylococcus aureus are the first organisms we're using this semester that are what we call opportunistic pathogens. And so we want to use proper aseptic technique. We want to wear the proper personal protection equipment. And of course, we want to wash our hands, sanitize our hands when we're done, and use caution when handling these as they are potentially pathogenic. Now, when you stab the tubes today, you're only going to stab the bacterium about 1/2 to 3/4 of an inch into the agar. And it's very important that when you stab the agar, you take care not to tilt or twist the loop so the loop comes up through the same cut as it went down. We want to get a nice straight line down, a nice straight line up, because any place you cut into the agar or any place you twist the loop, the bacteria are going to grow. And you don't want to confuse several inoculation areas in there where you twisted the loop or tilted it for spreading. That's simply where the loop cut into the agar. So we need a nice straight line when you inoculate it. And you're going to incubate those 2 tubes in your test tube rack at 37 degrees Celsius until next time. And finally, we're going to observe several flagella stain demonstrations. We have a slide of a Vibrio species, show you a monotrichous arrangement like we see here. We have Proteus vulgaris, which has a paratrichous arrangement, as we see in figure 9. So again, you'd recognize that arrangement as paratrichous. And we have a Spirillum species that shows you an amphitrichous arrangement, a single flagellum at both ends, so we'd recognize that arrangement as amphitrichous. And of course, you'll be recording your results. So for part A, the endospore stain, that's all demonstration. We have 3 slides we're looking at. Bacillus megatherium, done with a staining technique where the vegetative cell appears red and the endospores appear green. We have an endospore stain of Bacillus anthracis that causes anthrax, another streptobacillus. In this case, the vegetative bacteria stain blue, and the endospores are colorless within the bacillus. And we have an endospore stain of Clostridium tetani, where the vegetative cells stain a grayish blue, and the endospores are colorless. And again, if we see that tennis racket shape on the bacillus, we should recognize the genus as Clostridium. And remember, again, genus is the first of the two scientific names. We don't want the species name tetani. We can only tell the genus name Clostridium because of that shape, if we see it. Then you record the results of your motility test medium here and your conclusions as to whether it's modal or non-modal and how you can tell. And then make some drawings of the three flagella stains we're showing you monotrichous, paratrichous, and amphitrichous arrangements. And of course, always at the end of the lab, we have our performance objectives that tell you what you have to be able to do on the lab quiz covering this lab, and what you have to recognize under results for the practical. And of course, at the end of every lab, we also have a brief self-quiz and some answers for that self-quiz. So that's bacterial endospores and bacterial motility.